New molecular techniques link genes to DNA, RNA, and proteins. The modern synthesis of the 1940s took place prior to the revolution in molecular genetics that began with the discovery of DNA in 1944, but did not get underway until the discovery of the structure of DNA in 1953, the cracking of the genetic code in the 1960s, and the recombinant DNA revolution in the 1970s. Proving that DNA is the genetic material of living things. Prior to 1944, biologists suspected that proteins, rather than DNA, constituted the genetic material of living things. Both DNA and proteins are found in chromosomes, but proteins are far more complex since they consist of sequences of 20 different amino acids rather than 4 different nucleotides. Proteins seemed to be a better candidate for the genetic material because it seemed likely that genes would require a complex molecule to encode them. The definitive experiment that proved that DNA, not proteins, is the genetic material of living things was performed by Avery, McLeod, and McCarthy in 1944. The complex relationship between clinical and basic research. Oswald Avery spent most of his early career as a tireless and meticulous researcher who was determined to find the infectious agent that caused influenza. Influenza, complicated by bacterial pneumonia, was responsible for some 50 to 100 million deaths worldwide during the flu pandemic of 1918. Although the virus that causes influenza was not identified until 1931 by Richard Shope, Avery's research focused on a virulent bacterial strain, a secondary infection of flu victims that was responsible for the majority of deaths in the 1918 pandemic. The Great Influenza, an engaging popular historical account by John Barry, tells the clinical history behind the serendipitous discovery of the phenomenon known as genetic transformation by Oswald Avery, McLeod, and McCarthy. Briefly, they discovered that DNA, taken up by bacterial cells, could turn harmless strains of bacteria into lethal pathogens. This led to critical experiments demonstrating that DNA is the genetic material of all cells in 1944, resulting in the Nobel Prize. This illustrates the complex, mutually beneficial relationship between basic and clinical research. It also shows that luck and accidental discoveries play major roles in both basic and applied research, and the value of a particular line of research often cannot be predicted in advance. Biomedical science rests on a firm foundation of basic research, and it is necessary to cast a very wide net.